Hey everyone, Gina Legrand here. Thanks for taking the time to view my presentation about the Leucine Zipper. I'm hoping you'll find it informative and interesting. Our first slide gives us a brief overview of the Leucine Zipper. A leucine zipper, also known as a zip, is formed by amphipathic interactions between two alpha helices, one from each monomer. Amphipathic interactions are those caused by proteins having both hydrophobic and hydrophilic components. The helices are held together by hydrophobic interactions between leucine residues, which are located on one side of each helix. Because of the tight packing of these two hydrophobic side chains, the leucine zipper is a very stable structure. As you can see from the image, the red arrows indicate where the carboxyl group binds to an amino group 4 residues ahead in the chain. We refer to the second slide to explain what principles are involved in holding the two alpha helices together. The alpha helix is a secondary structural element that is seen frequently in proteins. To review, alpha helices are polypeptides stabilized by intrachain hydrogen bonds between the amino and carbonyl groups, creating a coiled backbone. Each CO group forms a hydrogen bond with an NH group that is situated 4 residues ahead in the chain. There are 3.6 amino acids per turn of the alpha helix. Leucine zipper domains consist of 2 motifs. A motif is a basic region that recognizes a specific DNA sequence and a series of leucines spaced 7 residues apart on the alpha helix, typically in a repetitive alpha helix turn helix conformation. This region interacts with DNA bases, which themselves are joined by hydrogen bonds. Most zip motifs form a parallel coiled structure, although they can be formed by 2 anti-parallel alpha helices. All helices have a screw sense, which describes whether the rotation of the helix is right-handed or left-handed. As we learned, helices found in proteins are almost exclusively right-handed because this conformation reduces steric clashes. The leucine zipper is a coiled coil, essentially 2 right-handed coils intertwined to create a left-handed superhelix. This type of structure is commonly found in alpha-keratin, a fibrous protein and a primary component of wool and hair. The helices in a coiled coil are held together by van der Waals and ionic interactions, and also by disulfide bonds formed by neighboring cysteine residues. When we look at the image, we can see that it takes 3.6 amino acid residues to complete a turn. Our third slide suggests one possible residue that could replace leucine and still maintain the hydrophobic core, as well as one that could potentially destabilize it. 2 possible amino acids that closely resemble leucine are valine or isoleucine, and due to their similar bulky structure might be effective substitutes. Residues that are too conformationally restricted produce interactions that cause steric clashes or increase the potential for electrostatic interactions could potentially destabilize the core. Replacing leucine with polar amino acids such as serine or threonine would create electrostatic interactions between the lysine and the OH groups on serine and threonine, dramatically changing the structure and destabilizing the core. You can see from the images that isoleucine and valine are structurally very similar to leucine, allowing the core to maintain its integrity. In contrast, you can see that the polar OH regions of serine and threonine could create unexpected interactions at both the carboxyl and amino ends of the backbone. The final slide in my presentation addresses finding a substitute for the amino acid pair lysine and glutamate. 2 other side chains that have similar structure to lysine and glutamate are arginine and aspartate. However, the polarity of arginine's guanidinium group could pose some unwanted electrostatic interactions between the carboxyl group of the aspartate and the amino group double bonded to carbon and asparagine, causing unwanted repulsion or altering how the alpha helices coil. The drawings indicate that asparagine and aspartate are very similar in structure to lysine and glutamate. However, the potential for unexpected interactions still exists between the 2 terminal NH groups and their resonant bond to carbon on asparagine, which restricts rotation. Thank you again for taking the time to view my presentation on the leucine zipper. I hope you enjoyed it. I look forward to viewing each of your presentations over the next few days.